This is Heliox, where evidence meets empathy. Independent, moderated, timely, deep, gentle, clinical, global, and community conversations about things that matter. Breathe easy, we go deep and lightly surface the big ideas. Imagine that scientists spend 3 decades and literally billions of dollars developing a drug to cure a completely devastating disease.
Yeah, an unbelievable amount of time and money.
Right. And they identify the villain. It's this massive, tangled clump of proteins that is just clearly visible in the brains of patients. So they spend years designing molecular compounds to dissolve this clump.
It was basically the holy grail of neurobiology at the time.
Exactly. And finally, they run the clinical trials. The drug works perfectly. I mean, it enters the brain, it finds the tangled clumps, and it breaks them apart.
Just like it was supposed to.
Just like it was supposed to. The whole scientific community is just holding its breath, and then the results come in. The clumps are gone, sure, but the patients are not getting better.
Right.
In fact, some of them are getting worse. The disease just continues its relentless march.
It was one of the most sobering moments in the history of modern medicine. Entire pharmaceutical departments, they had to go all the way back to the drawing board.
Just start over from scratch.
Yeah, totally. And it forced the scientific community to ask this really chilling question. What if destroying those giant plaques was actually destroying the brain's very last line of defense?
It gives me goosebumps just thinking about it. Hi, everyone, and welcome. Today, we are looking at the paper that explains exactly how we got it so wrong, and more importantly, how a new wave of researchers is getting it right.
I'm really excited for this one.
Me too. We are doing a deep dive into a really monumental piece of source material. It was published in the FVBS journal, and it's titled Proteostasis of Organelles in Aging and Disease.
Yeah, and this work comes from Yaron Abawi, Kansu Dosen, David Vilches, and their whole team over at the University of Cologne.
Right. And what they are laying out here is, well, it's nothing short of a total paradigm shift.
Oh, absolutely. The journey these researchers and really the broader field have taken over the last few years is just a master class in reevaluating our fundamental assumptions.
Because we had it so incredibly wrong for so long.
We did. For a long time, we viewed a human cell as essentially like a bag of enzymes, just a microscopic bowl of soup.
A bowl of soup, right.
Yeah. And when things went wrong in aging, we just kind of assumed the soup as a whole just went bad. It all just sort of expired at once.
Right, like milk left out on the counter.
Mm-hmm.
But the Vilches lab and the sources we are exploring today, they present this radically different reality.
Yeah, the cell is definitely not a uniform soup. It is exquisitely compartmentalized into these really highly specialized neighborhoods, which are the organelles.
So listener, by the end of this deep dive, you are going to understand the commonalities of how your individual cells actually age.
It's pretty mind-blowing stuff once you get down to the physics of it.
It really is. We are going to look at the microscopic physics that keep you alive every single second you are listening to this.
Yeah.
We'll look at the exact mechanisms of why those physics eventually fail.
And we'll get into the wild futuristic strategies being developed to extend human health span.
We are going to map the neighborhoods. But before we can take a tour of how these individual cellular neighborhoods break down in aging and, neurodegenerative diseases like Alzheimer's and Parkinson's, we need to establish the baseline.
Right. We have to understand the foundational physical challenge that every single cell in your body is trying to overcome at this exact moment.
Which means we have to talk about protein folding.
We do. Because, most of us learned about the genome in school, the DNA, the master blueprint.
Right, the double helix. We all know the picture.
Exactly. But the DNA is just information sitting in a vault that doesn't actually do any of the physical labor of keeping us alive.
So who does the heavy lifting?
The physical labor is done by the proteome. The proteome is the entire set of proteins expressed by a cell at any given time.
And there are a lot of them, right?
A staggering amount.
Mm-hmm.
Human cells express over 10,000 different proteins simultaneously. These are the actual molecular machines.
So they're building things.
Yeah, they form the structural scaffolding of your cells. They act as enzymes to digest your food. They are the receptors that catch signals from your brain.
They're the antibodies fighting off infections too, right?
Exactly. If the genome is the architectural blueprint, the proteome is the concrete, the steel, the wiring, and the actual construction workers themselves.
I love that analogy. And a protein doesn't just pop out of the cellular factory fully formed and ready to do its job, does it?
Not at all. When the ribosome, which is the cell's protein factory, reads the genetic code, it prints out a protein as a long linear string of amino acids.
Like a piece of yarn.
Yeah, like a really long piece of yarn. But a piece of yarn can't act as a highly specific enzyme or sturdy structural beam. It has to fold.
And the folding process is, well, it's one of the most fascinating phenomena in biophysics, I think.
Oh, completely. The linear string must fold into a very specific, intricate, three-dimensional shape. We call this its native conformation.
And that shape dictates what it does.
Its shape dictates its function entirely. If it folds incorrectly, it's useless. Or, even worse, it's actively dangerous.
So how does a blind microscopic string of amino acids just know how to fold into a complex 3D shape? I mean, there's no tiny microscopic architect bending it into place.
No, there isn't. It is driven entirely by the laws of thermodynamics.
Okay, break that down for us.
So, the sequence of amino acids contains some molecules that are hydrophilic, meaning they readily interact with water, and others that are hydrophobic, meaning they're chemically repelled by water.
They hate water.
I absolutely hate it.
Yeah.
And because the inside of a cell is an aqueous, watery environment, that newly made protein string is immediately subjected to intense physical forces.
So the water is actually pushing on it.
Yeah, the water molecules essentially push and pull at it. The protein naturally and spontaneously contorts itself to seek the lowest possible free energy state.
Meaning it tries to hide the parts that hate water.
Exactly. It violently twists to bury all of its hydrophobic, water-repelling parts deep on the inside of its own structure, while keeping its water-loving parts exposed on the surface.
So it's hiding the parts of itself from the environment. That actually sounds like a fairly elegant, self-solving physical equation.
And it would be, if it were happening in a vacuum or just a perfectly calm pool of water.
Mm-hmm.
But this is where our mental model of the cell usually fails us.
Right, the textbook drawings.
Yeah, textbooks often illustrate the cytosol, the fluid inside the cell, as this spacious, serene environment where a few organelles just float around peacefully.
Like a lava lamp or something.
Right, a lava lamp.
Yeah.
But the physical reality of the cytosol is unimaginably harsh and violent.
How crowded are we actually talking here?
The concentration of proteins in the cytosol is roughly 300 grams per liter.
300 grams? That is incredibly dense. That's significantly thicker than gelatin, isn't it?
It is. It's not really a fluid. It's more like a vibrating gel.
A vibrating gel. Wow.
And everything in that gel is moving constantly. Because of thermal energy, these tens of thousands of proteins and molecules are vibrating, spinning, and just crashing into each other at breakneck speeds.
That sounds chaotic.
It's totally chaotic.
Yeah.
In physics, this is known as the excluded volume effect. There is virtually no free space in there.
Okay, so trying to fold a complex protein in that environment, it's like standing in a packed, violently swaying subway car at rush hour, and you're trying to fold an intricate, multi-layered origami swan without your elbows bumping into anyone else.
That is the perfect way to visualize it. And remember those hydrophobic parts of the protein string that are trying to hide from the water.
Right, the sticky parts.
Exactly. While the protein is in the process of folding, those hydrophobic patches are temporarily exposed to the environment, and they're incredibly sticky.
So what happens if they bump into something else on the subway?
Well, if the exposed hydrophobic patch of one unfolding protein crashes into the exposed hydrophobic patch of another protein on that crowded subway, they will instantly bind together.
Just to escape the water.
Yes, exactly. And this is called aggregation. The moment they aggregate, neither protein can finish folding. You suddenly just have a tangled, useless clump of molecular garbage.
So you need help on the subway. You need some kind of buffer zone.
Which brings us to the proteostasis network, or the PN. This is the maintenance crew of the microscopic metropolis.
A PN.
The proteostasis network is this massive, highly coordinated system of roughly 2,000 different factors, and they are all working together to maintain the integrity of your proteome.
And who are the frontline workers of this network?
The frontline workers are called the chaperones.
Chaperones, like at a middle school dance, making sure the teenagers don't get inappropriately tangled up.
The scientific nomenclature is rarely that perfect, but yeah, in this case it really is. Chaperones are specialized proteins whose sole evolutionary purpose is to assist other proteins in folding without interacting inappropriately.
That's amazing. What kind of chaperones are we talking about?
The sources detail several major classes, things like HSP70, HSP90, and this massive complex called the T-right chaperonin.
So how do they actually do it? If they're floating in the exact same crowded subway car, How do they stop the sticky hydrophobic patches from clumping together?
Let's look at HSP70, which is one of the most ubiquitous chaperones in our cells. It has a physical structure that acts somewhat like a molecular clamp.
A clamp, okay.
Yeah. When a new protein is emerging from the ribosome, HSP70 recognizes those exposed hydrophobic patches. It literally physically binds to them, covering them up like a shield.
So it's hiding the sticky parts.
Exactly. It uses cellular energy ATP to clamp down super tight. By covering the sticky parts, HSP70 prevents the protein from aggregating with its neighbors.
Giving it the safe space and the time it needs to find its correct internal fold.
You got it. And once the protein is fully folded, the hydrophobic patches are safely buried inside naturally, and the chaperone realizes its job is done and releases its grip.
That implies just an incredible evolutionary history. I mean, this isn't just some modern biological luxury. This feels like a fundamental requirement for complex life to even exist.
Oh, absolutely. If you look back through the evolutionary tree, the basic principles of this network trace all the way back to simple bacteria.
They have chaperones too.
They do. They use a very basic chaperone called trigger factor. But as life evolved into complex eukaryotes, like us, like human beings, our proteomes became massively larger and way more complicated.
Right, because we have proteins that are thousands of amino acids long.
Exactly. And with multiple distinct domains that all have to fold in a very specific sequence, a simple clamp just wasn't enough anymore.
So what did evolution do?
Evolution built vast hierarchical networks of what we call co-chaperones. These are basically helper molecules that regulate the main chaperone.
Like managers for the frontline workers.
Yeah, exactly. They manage the flow of cellular energy. They dictate exactly when the chaperone should clamp down and when it should let go. They even pass misfolded proteins from one chaperone system to another like a microscopic assembly line until the job is done perfectly.
It's a whole logistical empire. But the proteostasis network isn't just about folding new proteins, is it? It has to manage the entire life cycle of a protein.
Right, because proteins naturally get damaged over time.
So it's a delicate three-part balancing act.
Exactly. First, you have synthesis of the ribosomes. Second, you have the folding and conformational maintenance handled by the chaperones we just talked about. And third, you have degradation.
Waste disposal.
Critical waste disposal. If a protein is damaged beyond repair by heat or oxidative stress, or if it has simply served its purpose and is no longer needed, it must be destroyed.
To prevent clutter in that already insanely crowded cell.
Exactly. And the cell uses two main systems for this. The first is the ubiquitin proteasome system, or the UPS.
How does the UPS work? I'm picturing little brown delivery trucks.
It's actually more like a molecular tagging system. When a protein is recognized as irrevocably misfolded, special enzymes attach a chain of small proteins to it. These small proteins are called ubiquitin.
Ubiquitin. Okay.
Think of ubiquitin as a microscopic barcode that just screams trash.
I love that. A trash barcode.
Yeah, and once a protein has this barcode, it is routed to the proteasome. The proteasome is essentially a highly structured molecular wood chipper.
A wood chipper. Inside the cell.
It pulls the tagged protein inside its barrel-like structure and chemically chops it back down into individual amino acids.
Which the cell can then recycle to build brand new protein.
Exactly. It's a completely closed-loop recycling system.
That is so efficient. And what is the second degradation pathway?
That would be autophagy. While the UPS handles individual proteins, autophagy acts more like a cellular recycling center for much larger clumps,
Like what kind of clumps?
like aggregated proteins or even entirely damaged organelles. Autophagy involves building a double membrane structure called an autophagosome.
And what does that do?
It physically engulfs the large biological trash, seals it up, and then fuses with the lysosome. And the lysosome is full of highly acidic enzymes that just burn the trash down to its base components.
Okay, so we have this magnificent, highly evolved, intricately balanced maintenance crew. It has been perfected over literally billions of years of evolution.
It really is a marvel.
It handles synthesis, it handles folding, and it handles degradation. It prevents the crowded subway from turning into a massive pileup.
Right.
Which brings us back to the agonizing question we started this whole deep dive with. If this system is so perfectly tuned, why does it fail so spectacularly?
Yeah.
Why do we get devastating neurodegenerative diseases like Huntington's, Alzheimer's, and Parkinson's late in life?
Well, answering that question forces us to confront the massive scientific dead end that we discussed at the very beginning of the show.
The failed drugs.
Exactly. To understand the paradigm shift made by researchers like Nabawi, Dosan, and Vilches, we have to look closely at the trap that caught the entire scientific community for decades.
And this trap has a name, right? The amyloid hypothesis.
Yes, the amyloid hypothesis. Back in the early 20th century, Alois Alzheimer examined the brain of a deceased patient who had suffered from severe dementia.
And what did he see?
Under the microscope, he saw something glaringly obvious. The brain tissue was just littered with massive insoluble fibrillar protein aggregates.
Just giant clumps of tangled, misfolded proteins.
We now know them as amyloid plaques in Alzheimer's or Lewy bodies in Parkinson's.
And because these massive plaques were so visually striking, and because they were always found at the scene of the crime in neurodegenerative brains, the scientific community logically concluded that these large aggregates were the primary cause of the cell death.
It is a textbook case of confusing correlation with causation, though, to be fair, it is an incredibly understandable one.
Right. I mean, if you see a giant, rigid clump of garbage crushing the delicate structures of a neuron, you naturally assume the garbage killed the neuron.
Of course. And the entire field rallied around this concept. The strategy became super simple in theory. Figure out how to dissolve the garbage, and you save the brain.
This triggered an absolute gold rush in the pharmaceutical industry, didn't it?
Oh, massive. Billions of dollars were poured into developing anti-aggregation therapies. Researchers engineered these brilliant molecular tools,
Like what?
like highly specific antibodies designed to cross the blood-brain barrier, bind to these massive amyloid aggregates, and physically break them apart or signal the immune system to clear them out.
And they succeeded in doing that, didn't they? The drugs did exactly what they were designed to do.
They did. That's the tragedy of it. In many clinical trials, imaging showed that the drugs successfully cleared the massive plaques from the brains of the patients.
But the cognitive decline continued unabated.
Unabated. The patients still lost their memories, their motor control, and eventually their lives. The sources we're looking at note that despite thousands of therapies developed in the lab, no disease-modifying therapy targeting these large aggregates succeeded clinically in stopping the disease.
It's just heartbreaking. So what went wrong? If the massive clumps of misfolded protein weren't killing the cell, what actually was?
Well, we had the villain completely wrong. The sources reveal a fascinating, if tragic, twist. Those large amyloid aggregates, the giant plaques we spent 30 years trying to destroy, are not the primary toxic species.
They aren't?
No. In many ways, they might actually be a protective mechanism enacted by a desperate cell.
Wait, hold on. So the giant, scary plaques we've been trying to eradicate for decades were actually the cell's attempt to build a quarantine zone.
Essentially, yes.
We were attacking the cellular equivalent of sandbags during a flood.
Precisely. We were blowing up the sandbag.
Oh, my God.
To understand why the cell builds these massive plaques in the first place, we have to look at the real villains. They are called soluble oligomers.
Soluble oligomers. Okay, what is an oligomer?
When a protein first misfolds, before it becomes a giant solid plaque, it exists as a small, free-floating cluster of maybe a dozen or a few hundred proteins.
Okay, so a tiny clump.
Right. And because this cluster is small, it remains soluble in the water of the cytosol. It can float freely throughout the cell. And because it is misfolded, it still has those intensely sticky, hydrophobic patches exposed on its surface.
Ah. So you have these tiny, invisible, incredibly sticky clusters just floating freely around the delicate machinery of the cell. Why are they so much more toxic than the massive plaques?
Their toxicity comes from their mobility and their exposed surface area. Because they are floating freely, they inevitably encounter the delicate lipid bilayer membranes of the cell and its organelles.
The walls of the compartments.
Exactly. Lipids have hydrophobic tails. The exposed hydrophobic patches on these floating oligomers are drawn to the lipid membranes. They physically embed themselves right into the membrane, forcing the lipids apart.
They punch holes in the membrane.
They do. They act like unregulated ion channels. Suddenly, the tightly controlled barrier of the cell is completely compromised.
And what happens when a hole gets punched in the cell membrane?
Well, calcium ions, which are strictly regulated, flood into the cell through these new pores. This massive influx of calcium acts as a catastrophic distress signal, eventually triggering apoptosis, which is programmed cell death.
That is horrifying. A bunch of microscopic hole punchers.
But it gets worse, doesn't it? Because these oligomers don't just attack the membranes, they attack the maintenance crew itself.
They do. Because
Okay.
the oligomers have these exposed, sticky patches. The chaperones, like HSP70, they recognize them as misfolded proteins.
Right, because that's their job.
Exactly. So the chaperones clamp down on the oligomers, trying to help them fold. But the oligomers are fundamentally broken. They cannot be folded. So what happens to the chaperone?
It just stays clamped. They get permanently permanently glued to the toxic oligomers.
Wow. The maintenance crew is essentially taken hostage.
Taken hostage, exactly. And this rapidly depletes the pool of available chaperones in the rest of the cell.
Hmm.
Suddenly, normal, healthy proteins that are just being synthesized have no chaperones available to help them fold.
Oh, no. So they start misfolding, too.
Yes, they start misfolding, creating more oligomers, taking more chaperones hostage. It's an exponential cascade of failure.
Okay, so if the cell is rapidly filling up with these highly toxic, hole-punching, chaperone-hijacking oligomers, what does it do? How does it fight back?
It initiates a desperate physical containment strategy. It literally sweeps all these small, free-floating oligomers into a single, massive pile. It forces them to bind together into a dense, insoluble, fibrillar aggregate, a plaque.
Why does binding them into a massive plaque help, though?
It is a matter of simple geometry, surface
Yeah.
area to volume ratio.
Okay, math time.
Yeah, think about it like this. If you have 10,000 small oligomers floating around, they have a massive amount of exposed surface area interacting with the cell.
Right, lots of sticky edges exposed.
Exactly. But if you pack all 10,000 of them into one giant sphere, you drastically reduce the total exposed surface area. The sticky, toxic parts are safely buried inside the massive plaque, completely sequestered away from the membranes and the chaperones.
So when our billion-dollar drugs went in and busted up those large plaques...
We were essentially detonating the quarantine zone.
Wow.
We were breaking the massive inert plaques back down into thousands of highly toxic free-floating oligomers, releasing them back in the wild to punch holes in the cell.
It is a complete inversion of how we thought about the disease. And it explains why treating the entire cell as one big homogeneous problem just wasn't working. We couldn't just throw a drug in there to scrub away the visible garbage.
Right. Which brings us all the way back to the central thesis of the FBS journal paper. Recognizing the massive failure of the amyloid hypothesis, researchers like Nabawi, Dozan, and Vilches took a huge step back.
They looked at the big picture.
They realized that treating the cell as a simple bag of enzymes, a uniform soup, was totally blinding us to how proteostasis actually works.
So they shifted the paradigm from the soup model to the compartment model.
Yes. Their core argument is that proteostasis is exquisitely compartmentalized. Human cells have evolved distinct membrane-bound organelles. The nucleus, the endoplasmic reticulum, the mitochondria, the lysosomes.
The different neighborhoods.
Exactly. Historically, biology textbooks treated these membranes just as structural dividers, kind of like walls in a house. But the researchers highlighted that these membranes exist to create unique, highly specialized biochemical microenvironments.
Okay, what makes an environment biochemically unique?
Things like pH levels, calcium concentrations, and redox states. For example, the inside of a lysosome has a highly acidic pH because its whole job is designed to dissolve biological material.
Right, it's a stomach.
Basically, yeah. But the inside of the endoplasmic reticulum is a highly oxidizing environment to allow for specific chemical bonds to form, whereas the general cytosol outside is a reducing environment.
So we're talking about different neighborhoods with completely different atmospheres.
Exactly. And because these environments are so biochemically distinct, the physical rules of protein folding change in each one.
Oh, that makes sense.
Right. A chaperone that evolved to function perfectly in the highly acidic soup of a lysosome would instantly denature and fail in the alkaline environment of the cytosol. Therefore, you just cannot apply a one-size-fits-all maintenance strategy to the cell.
It's kind of like a city realizing it can't use the exact same maintenance strategy for a pristine art museum that it uses for a heavy-duty steel mill.
Oh, I like that.
Right. The art museum needs delicate climate control and fine dusting, but the steel mill needs heavy machinery and heat shielding.
That is the perfect analogy.
Yeah.
If you try to use the art museum cleaners in the steel mill, they will fail instantly. Each organelle requires its own tailored protein quality control system, its own specific set of chaperones and degradation pathways calibrated to its exact biochemical atmosphere.
This fundamentally redefines what aging actually is at a cellular level, doesn't it?
It completely redefines it. Aging is no longer viewed as a generalized global slowing down of the entire cell. According to this research, aging is the progressive, localized disruption of these specific organelle microenvironments.
So the art museum's climate control might fail long before the steel mill's machinery breaks down.
Exactly. To understand how a cell dies, you have to look closely at which specific neighborhood's maintenance crew goes on strike first and why.
Well, I definitely want to take that tour. I want to
Yeah.
walk through these cellular neighborhoods and look at the exact mechanisms of how they break down as we get older. Let's start at the command center, the nucleus.
Let's do it. So the nucleus is the vault that houses your DNA, but it is also an incredibly active workspace. It is highly crowded with what we call metastable proteins.
Metastable, what does that mean?
It means they are proteins that are very sensitive to environmental changes and are highly prone to misfolding.
Okay, but if the proteins managing your DNA misfold, that sounds like an immediate catastrophic failure.
It would be, but the nucleus has evolved a fascinating architectural trick to prevent that. Deep within the nucleus, there is a dense, distinct region called the nucleolus.
The nucleolus, I remember that from high school biology. We learned it was where ribosomes are made.
Right. Traditionally, we thought of the nucleolus just as the place where ribosomes are assembled. But the sources describe a secondary, totally vital role. During times of acute cellular stress, like a sudden spike in body temperature from a fever, which causes proteins to start unraveling.
Oh, right. Heat unfolds proteins.
Exactly. When that happens, the nucleolus acts as a phase-separated recovery room.
Phase separated, like oil and water.
Yes, exactly. The nucleus isn't actually bounded by a membrane. It maintains its structure purely through the physical interactions of the molecules inside it, kind of separating from the rest of the nuclear fluid.
That's wild. So what happens during a fever?
When a heat shock hits, misfolded nuclear proteins are actively shuttled into this dense nucleolar droplet. They are transiently sequestered there, kept completely safe from aggregating with the delicate DNA machinery until the danger passes.
It's literally an emergency bunker. The sirens go off, the proteins run into the bunker, and they wait out the bombing.
Yep. And once the fever breaks and the stress is over, nuclear chaperones pull those proteins back out of the nucleolus and refold them. It's a beautifully elegant, dynamic system.
But I sense a but coming.
But here is where aging enters the picture.
Mm.
With age, the dynamic nature of this recovery room is compromised.
The bunker doors get jammed.
Precisely. Due to a slow decline in cellular energy and chaperone capacity over decades, the misfolded proteins still go into the nucleolus when stress hits, but the system loses the ability to pull them back out. Yeah. The transient recovery room solidifies into a permanent prison. The proteins aggregate inside the nucleolus, disrupting its ability to make ribosomes, and nuclear function begins an irreversible decline.
So the command center locks itself down and basically slowly suffocates. If the nucleus is failing, what happens to the rest of the city? What happens to all the proteins that need to be manufactured and shipped out?
That brings us directly to the industrial district, the endoplasmic reticulum.
The ER.
The endoplasmic reticulum, the ER, is the cell's massive mass manufacturing and folding factory. Roughly 1/3 of all the proteins your cell makes are manufactured, folded, and quality checked inside the ER.
A third. That's huge, especially the ones destined to be secreted into the bloodstream, like hormones or embedded in the cell membranes, right?
Exactly. That is an immense volume of protein processing. It's a continuous high-speed assembly line.
And because the volume is so high, I imagine the ER is incredibly sensitive to even the slightest delay.
Oh, incredibly sensitive. If proteins start misfolding, they accumulate rapidly. To prevent a catastrophic pileup, the ER relies on a brilliant, highly complex surveillance system called the Unfolded Protein Response, or UPR.
UPR. How does the ER know when things are piling up, though? It doesn't have eyes.
No, it uses 3 specific transmembrane stress sensors that are built physically into the wall of the ER. They're called IRE1, PERK, and ATF6.
Okay, IRE1, PERK, and ATF6. How do they work?
These proteins sit with one end inside the ER lumen, constantly monitoring the folding environment, and the other end sticking out into the general cytosol, ready to signal the rest of the cell.
So they're like sentries on the factory wall. Let's focus on the mechanism here. How do these sensors actually react when they detect a pileup of misfolded proteins inside?
Let's look at the sensor PARC as an example. When chaperones inside the ER get overwhelmed by misfolded proteins, they physically detach from the inside end of PROOK.
Because they have to go deal with the garbage.
Exactly. They get pulled away. And this detachment causes PERK to activate. The end of PERK that sticks out into the cytosol acts as a kinase.
A kinase. Remind me what that is.
A kinase is an enzyme that attaches phosphate groups to other molecules. It's a way of chemically modifying them.
So PERK reaches out into the cytosol and starts attaching phosphate groups to what exactly?
It physically phosphorylates a critical translation factor called EIF2 alpha.
Okay, EIF2 alpha. What does that do?
Well, normally EIF2 alpha is essential for the ribosome to start building a new protein. But when PERK attaches that bulky, negatively charged phosphate group to it, EIF2 alpha's shape changes. It can no longer bind to the ribosome.
It throws a physical wrench into the gears of the factory.
Exactly. By phosphorylating eIF2-alpha, PERK initiates what is called translation attenuation. It completely halts the synthesis of almost all new proteins entering the ER.
It literally hits the stop the assembly line button.
It does. And this is a brilliant survival mechanism because it stops the influx of new proteins, giving the overwhelmed chaperones inside the ER the time they need to clear the backlog and restore order.
Okay, I follow the logic. It's a great failsafe. But wait, if the ER hits the emergency stop button on protein production and halts the synthesis of a third of the cell's proteins, doesn't the cell need those proteins to live?
That is the profound, tragic compromise of aging.
I mean, if you stop making membrane receptors and structural proteins, doesn't the cell eventually just starve to death?
Yes. In a young, healthy cell, hitting that stop button is a temporary, highly effective measure. The backlog is clear, the chaperones return to PERK, the phosphate group is removed from eIF2-alpha, and production happily resumes.
But in an older cell.
As we age, the cell faces chronic, unrelenting stress. The misfolded proteins keep piling up way faster than they can be cleared, and the alarm just stays on too long. The assembly line remains shut down.
So the safety mechanism itself becomes the executioner.
It does. And the sources reveal an even more fatal age-related flaw regarding the ER.
It gets worse.
It gets worse. As we get older and the ER faces this chronic protein misfolding stress, it actually undergoes a physical, structural remodeling. It fundamentally shifts its primary function.
What does it do?
It downregulates the genes responsible for protein folding and quality control, and instead upregulates genes responsible for lipid processing and cholesterol synthesis.
Wait, it just gives up. It changes industries entirely. Why on earth would it do that?
Because the cell is desperately trying to survive the immediate crisis. Chronic protein misfolding is wildly energetically expensive, and it creates massive amounts of toxicity that physically damages the ER membrane.
Ah, so the factory walls are breaking down.
Right, so by shifting to lipid synthesis, the ER is essentially trying to patch its own failing structural integrity. It is prioritizing the immediate survival of the membrane over the long-term need for folded proteins.
It's like a factory manager realizing the product assembly line is an unsalvageable disaster, so they fire all the assembly workers, sell the machinery, and turn the building into a storage warehouse just to keep the bank from foreclosing.
That is exactly it.
It solves the immediate chaos, but the business is fundamentally dead.
A very accurate analogy. By abandoning protein quality control to focus on lipids, the ER dooms the cell. The misfolded proteins accumulate entirely unchecked now, the assembly line never restarts, and eventually the massive cellular distress triggers apoptosis.
Okay, so the command center is locked in a bunker. The industrial district has fired its workers and become a warehouse. Let's look at the power plants, the mitochondria.
Right, the power plants. Mitochondria are completely unique organelles. Because of their evolutionary history, they were once independent bacteria that were engulfed by our cellular ancestors. They have their own double membrane and their own small genome.
They're like a cell within a cell.
They are. But they still rely heavily on the rest of the cell. They must import over 1,000 different proteins that are encoded by the cell's main nucleus and synthesized out in the cytosol.
How do you get a massive protein from the cytosol through two distinct mitochondrial membranes and into the center of the power plant? That sounds like a logistical nightmare.
It is. They have to pass through narrow, highly specialized protein channels in the membranes. These are known as the Tim and Tom translocase complexes.
Tim and Tom?
Yeah, but here's the mechanical catch. The channel is extremely narrow. A fully folded, three-dimensional protein cannot physically fit through it.
So it has to be threaded through like a piece of yarn through a needle.
Exactly. Chaperones in the cytosol must keep the newly synthesized mitochondrial protein in a completely unfolded linear state as it is fed through the Tim and Tom pores.
And then what?
Once it gets inside the mitochondria, a completely different set of mitochondrial chaperones grab it and fold it into its final functional shape.
Okay, so what happens if the cytosolic chaperones are distracted, say, by those toxic oligomers we talked about earlier, and the mitochondrial protein accidentally folds before it gets to the pore?
Well, if it folds prematurely, it physically jams the translocase channel. It clogs the intake valve. Suddenly, the mitochondria can't import the proteins it needs to maintain its energy-producing machinery.
It suffocates. And to clear these jams and to handle proteins that get damaged by the intense heat and oxidative stress of energy production, the mitochondria must have their own internal waste disposal. right? I mean, they can't use the cell's main proteasome wood chipper.
Correct. They rely on their own specific internal proteases. Think of them as localized garbage disposals. One of the most critical is a protease called LON P1.
Lon P1.
Yeah. And similarly, another organelle called the peroxisome, which handles toxic waste processing and lipid metabolism, relies on a related protease called LON P2.
And based on the incredibly depressing theme of this tour so far, I am going to deduce that Lon P1 and Lon P2 do not age gracefully.
They definitely do not.
Uh,
During the aging process, for reasons we're still entirely mapping out, the expression and functional efficiency of these specific mitochondrial and peroxisomal proteases sharply decline.
so the localized garbage disposals break down.
Exactly.
What is the mechanical result of that breakdown?
Damaged proteins start to accumulate inside the power plants. Specifically, the proteins that make up the electron transport chain, which is the actual machinery that generates cellular energy, they become damaged and misfolded, but they aren't cleared away.
So the machinery just gets clunky.
It becomes incredibly clunky and inefficient. And when the electron transport chain is inefficient, electrons start to leak out of the intended pathway.
Leaking electrons inside an oxygen-rich power plant? That sounds like a recipe for a massive fire.
It creates a massive biological fire. Those stray electrons prematurely interact with oxygen molecules, creating reactive oxygen species, or ROS.
Free radicals?
Yes, free radicals.
We hear about free radicals all the time in like skincare commercials, fight free radicals, but what are they actually doing at a molecular level?
So a superoxide radical is a highly unstable molecule that is missing an electron. It desperately wants to be stable, so it will violently rip an electron away from the nearest molecule it touches.
Anything nearby.
Anything.
Yeah.
It will rip electrons off the mitochondrial DNA, mutating it. It will rip electrons off the delicate lipid membrane, destroying its structural integrity. It is a vicious, self-amplifying cycle of oxidative destruction.
The power plant is essentially melting down from the inside.
Exactly. It's a microscopic Chernobyl.
Okay, this tour paints an incredibly grim picture of cellular decline. The nucleus bunker jams, the ER assembly line shifts to a warehouse, and the mitochondrial power plants melt down and spray toxic waste.
It's a lot, I know.
But the researchers didn't stop at the membrane-bound walled factories, did they?
No, they didn't. Because while the walled neighborhoods are failing, researchers discovered an even stranger, much more dynamic phenomenon happening in the wide-open spaces of the cytosol.
Okay.
They discovered organelles without any walls at all.
Membraneless organelles. You know, when I first read that in the sources, I thought it was a typo. How can you have an organelle, which is a distinct cellular compartment, without a membrane? It's like having a room without walls.
I know, it totally defies our macro-level intuition, but it relies on incredibly elegant microscopic physics. We are talking about structures like stress granules, processing bodies, and those nuclear speckles we touched on earlier.
How do they stay together without a wall?
They're formed through a thermodynamic process called liquid phase separation, or LLPS.
Liquid phase separation. Let's break down the physical mechanics of that.
The easiest macroscopic analogy is mixing oil and vinegar in a salad dressing.
Okay, I can picture that.
If you input energy by shaking the bottle, they mix into a cloudy emulsion. But if you just let it sit, the system naturally seeks a lower energy state. The oil molecules prefer to interact with each other rather than the water and the vinegar, so they spontaneously demix.
Right, they separate and form distinct floating blobs.
Exactly. Highly concentrated oil droplets suspended in the surrounding fluid. Membraneless organelles do exactly this, but with proteins.
What kind of proteins can act like oil in a watery cell, though?
Proteins that contain what are called intrinsically disordered or low-complexity sequences. Remember earlier how we said proteins must fold into a rigid 3D shape to function?
Yeah, the origami swan.
Well, that isn't entirely true. Large segments of many proteins don't fold at all. They remain floppy, flexible, and highly dynamic.
Like wet spaghetti.
Exactly like wet spaghetti.
Mm-hmm.
Because they don't have a rigid shape, these disordered regions can rapidly form very weak temporary physical bonds with other disordered proteins, things like pipey stacking or electrostatic interactions.
So they just lightly hold hands.
Right. And when the cell undergoes severe stress, say a sudden viral infection or exposure to a nasty toxin, these floppy proteins suddenly seek each other out.
To protect themselves.
Through these weak multivalent interactions, they spontaneously condense out of the watery cytosol to form highly concentrated viscous liquid droplets.
They just materialize out of the fluid. What is the biological purpose of these droplets? Why do they do it?
Protection and pausing. During a crisis, the cell needs to halt normal operations and focus entirely on defense. These phase-separated droplets, known as stress granules, act like molecular sponges.
What are they sponging up?
They selectively trap precious messenger RNAs,
Yeah.
the instructions for building proteins, and pull them completely out of circulation. They safely harbor this genetic material inside the droplet, pausing its translation, while the rest of the cell deals with the viral attack or the toxin.
Wow, so the cell uses fluid dynamics to conjure these temporary floating life rafts to save its genetic material during a storm. That is breathtakingly brilliant.
It really is a stunning biological innovation.
Huh.
And they are meant to be highly dynamic. They assemble in minutes when the storm hits, and the weak bonds allow them to dissolve rapidly back into the cytosol as soon as the storm passes.
But this incredible innovation comes with a massive evolutionary vulnerability, doesn't it?
It does.
Let me guess. The life rafts don't dissolve.
Exactly. The sources highlight that up to 75% of signaling molecules in mammalian cells contain these unstructured, low-complexity regions. That is a massive portion of our cellular communication network relying on this delicate, wet spaghetti physics.
And the tragedy of aging occurs right inside these droplets.
It does.
What goes wrong mechanically? Why do they stop dissolving?
It comes down to thermodynamics again. The liquid droplet state is a delicate equilibrium. The proteins inside are highly, highly concentrated. If they are held in this highly concentrated state for too long due to chronic age-related stress where the storm never passes, or due to inherited genetic mutations that make the weak bonds slightly too strong.
The physics shift.
The physics shift.
Yeah.
Over time, those floppy, unstructured protein segments will naturally seek a lower energy, more stable conformation. They will physically transition from a disordered tangle into rigid, highly ordered beta sheet structures.
They crystallize.
They undergo an aberrant phase transition. The fluid dynamic liquid droplet essentially solidifies. It turns into a rigid, toxic, fibrillar aggregate.
So if the storm lasts too long, the liquid life rafts literally turn to concrete and sink the ship.
That is precisely what happens.
Yeah.
And this specific transition from a protective liquid droplet to a solid amyloid-like aggregate is now understood to be the direct molecular mechanism behind devastating neurodegenerative diseases.
Which ones?
Most notably ALS, or Lou Gehrig's disease, and certain frontotemporal dementias. Mutations in specific proteins like FUS or TDP43 drastically accelerate this liquid-to-solid transition. The very mechanism designed by evolution to protect the cell solidifies and destroys it.
Man, okay, I am truly grasping the immense scope of this problem now. We have local failures happening everywhere. The ER warehouse shuts down, the mitochondrial power plants leak radiation, the liquid life raft turn to concrete.
It's a systemic breakdown.
But here is the piece of the puzzle I am struggling to connect. We have trillions of cells. How does a local failure in one tiny liquid droplet or one microscopic mitochondrion in one cell cause a whole macro-level human being to age, lose their memory, and succumb to disease?
Right, how does it scale up?
Yeah. If one cell's ER fails, why doesn't the body just trigger apoptosis, kill off that one bad cell, clear the debris, and let the healthy cells around it take over?
That is perhaps the most profound question in the field of aging right now. And it brings us to one of the most cutting-edge areas of research discussed in the EVBS journal paper. It's a field called organelle contactology.
Contactology, the study of how organelles physically touch.
Exactly. For decades, when you looked at a textbook diagram, the organelles were drawn like isolated islands, just floating independently in a sea of cytosol.
Right, completely separate.
But with the advent of modern high-resolution electron microscopy, we now know that image is completely false. Organelles are not isolated, they are intimately tethered together.
Oh, wow.
They physically touch, interact, and communicate with each other through specialized bridging structures called membrane contact sites.
So they are physically holding hands.
They are holding hands constantly. And while this allows them to share resources efficiently, it also means that a failure in one is immediately broadcast to the others. The researchers describe a vital interaction network called the redox triangle.
What makes up the triangle?
The physical and chemical interplay between the mitochondria, the endoplasmic reticulum, and the peroxisomes. They are physically tethered together to coordinate lipid metabolism and handle oxidative stress.
Okay, but because they are connected.
Right, because they are connected. If the mitochondria's proteases fail, and it starts generating those toxic reactive oxygen species, that oxidative fire doesn't just stay in the mitochondria.
It travels across the bridges.
It propagates instantly along the physical contact sites directly into the ER and the peroxisomes. The ROS floods into the ER, oxidizes the delicate folding chaperones, and instantly destabilizes the ER's protein folding environment.
Triggering that UPR alarm we discussed earlier.
Exactly.
It's a microscopic domino effect. The power plant fire spreads directly to the factory floor.
Yes. And the research actually highlights a specific actionable biomarker of aging related to these contact sites.
A biomarker.
Yeah, there's a specific physical tether where mitochondria and lysosomes touch, and it's regulated by proteins called FIS1 and RAB7. In a healthy cell, they touch to transfer metabolites, and then they let go.
They unhold hands.
Right. But when this specific contact site fails to dissociate properly, when they hold hands too long and can't let go, it disrupts the function of both organelles.
It just gums up the works.
Exactly. And the sources point to this failure to dissociate as an early warning sign of cellular senescence. It's a biomarker that researchers can look for before massive neurodegeneration occurs.
That is incredible. But we are still talking about a domino effect within the boundaries of a single cell. I'm a listener sitting in my car or walking my dog. How does the failure of RAB7 in one of my cells take down my whole body?
Because the domino effect doesn't stop at the cell membrane. This is where the biology gets truly mind-bending. The researchers describe a phenomenon called cell non-autonomous regulation of proteostasis.
Non-autonomous, meaning the cell is regulating things outside of its own autonomy.
Exactly. If the endoplasmic reticulum in a specific cell in, say, your intestinal lining, experiences severe protein misfolding stress, it doesn't just suffer in isolation and die quietly.
It doesn't just trigger apoptosis and fade away.
No. The massive stress response triggers the release of systemic signaling molecules.
It shoots off a distress flare.
A literal chemical distress flare. The stressed gut cell secretes specific neuropeptides and utilizes pathways like Wnt signaling.
And where do those signals go?
These signaling molecules travel out of the gut into the bloodstream and circulate throughout the entire body. They can even cross into the central nervous system. A stressed cell in the gut can secrete a neuropeptide that physically binds to receptors on the neurons in your brain.
And what does the neuron do when it receives that distress signal from the gut?
The healthy brain cell, sensing this systemic panic, will actually alter its own internal proteostasis network in response. It might preemptively trigger its own unfolded protein response, shutting down its own translation to prepare for a perceived systemic crisis.
Wow! This completely upends how I view aging. It's not just a slow, gradual rusting of individual parts in isolation. It's a highly communicative, systemic panic cascade.
That's a great way to put it.
One physical tether fails between a mitochondrion and a lysosome. The redox triangle destabilizes, the ER catches fire, a chemical distress flare is shot out via neuropeptides into the bloodstream, and suddenly a biological alarm is ringing in the brain, telling perfectly healthy tissues to shut down their factories and panic.
It is a systemic collapse driven by an overactive, interconnected communication network. And this systemic collapse forces us to ask a profound evolutionary question.
Which is?
Why does it happen?
Right. If our bodies have the biological capacity to maintain perfect proteostasis, handle the subway crowding, and clear the garbage for 30, 40, or 50 years without a single hitch, why do we suddenly lose that ability?
Well, the answer might lie in a biological concept known as the disposable soma theory.
Disposable soma. Soma meaning the body. You were saying our bodies are evolutionarily disposable.
From the cold, calculating perspective of evolutionary biology.
Yes. Ouch.
I know, it's harsh, but the theory suggests that every organism has a finite, limited amount of energy available to it. It must divide that energy budget between two competing needs, maintaining its own physical body somatic maintenance and reproduction.
Okay, I see where this is going.
Evolution is driven entirely by passing on genetic information to the next generation. Therefore, once an organism has successfully reached sexual maturity, reproduced, and ensured the survival of its offspring, the evolutionary selective pressure drops basically to zero.
There is no evolutionary reason to keep the original organism perfectly maintained forever.
Exactly. Why spend immense amounts of biological energy perfectly maintaining the protein-folding chaperones of an 80-year-old human when that energy could have been spent decades earlier ensuring successful reproduction?
Wow.
The theory suggests that the abrupt collapse of the proteostasis network in adulthood is not an accident or a random mechanical failure. It is a programmed biological trade-off.
We deliberately cut the funding to the city maintenance crew because we diverted all the tax dollars to building the next city over.
That is the grim yet elegant evolutionary logic.
Wow. That is deeply unsettling, but logically flawless. But let's pivot away from our disposable nature because the Vilches lab and their peers didn't spend years mapping out this interconnected systemic collapse just to document our inevitable demise.
Absolutely not.
By understanding the specific pathways, the UPR, the chaperones, the phase separation, the organelle tethers, they have uncovered unprecedented therapeutic opportunities, right?
They absolutely have. This is where the paradigm shift from the amyloid hypothesis to organelle proteostasis pays massive dividends.
So what's the new strategy?
We are moving away from the failed reactive approach of trying to violently scrub away massive plaques after the damage is already done. The new era of therapeutics is focused on pharmacological adaptation of proteostasis.
Instead of fighting the symptoms, we empower the maintenance crew.
Precisely. We are looking at therapies that actively tune the network. For instance, researchers are developing specific small molecules, often called pharmacological chaperones.
Pharmacological chaperones, how do those work?
These are highly engineered chemical compounds designed to fit perfectly into the structural crevices of metastable proteins. They physically bind to the protein and stabilize its structure before it ever has a chance to misfold.
Wow.
Yeah, preventing those toxic, hole-punching oligomers from ever forming in the first place.
That is proactive medicine at the absolute molecular level. What about the ER? Can we fix the factory?
Well, remember how the ER uses the peer sensor to hit the stop button on translation, but chronic stress causes it to fail?
Yeah, the alarm stays on too long and the factory becomes a warehouse.
Right. Researchers are investigating compounds that can artificially modulate that exact response. For example, molecules that can gently prolong translation attenuation, giving the chaperones just a little more time to clear the garbage without the cell triggering the fatal switch to lipid production and apoptosis.
So we aren't trying to stop the subway from being crowded. We're just giving the chaperones better tools, better radios, and slightly longer red lights to manage the crowd flow safely.
That is a perfect summary. We want to empower the existing, highly evolved systems. We are also looking at drugs that pharmacologically activate the clearance systems.
You mean speeding up the trash disposal?
Exactly. Upregulating the UPS wood chippers and the autophagy pathways, so the cell's internal recycling centers run faster and more efficiently as we age.
And the sources point to some things that sound straight out of science fiction, organelle therapy.
Yes, we are entering the realm of cellular engineering. The text specifically discusses the concept of encapsulated mitochondria transplantation
Transplantation.
of an organelle. Yeah. If we know that the mitochondria in the brain of a Parkinson's patient are failing, their LONQ1 proteases are dead, and they are leaking massive amounts of ROS that triggers systemic collapse. What if we don't try to fix them?
What do we do instead?
What if we isolate perfectly healthy mitochondria from a different tissue, package them in protective liposomes, and deliver them directly into the diseased neurons to replace the failing power plants?
You could theoretically swap out the broken engines without replacing the car.
It is being actively explored right now. Another incredibly futuristic concept is degron-mediated proteostasis regulation.
Okay, what is a degron?
A degron is a specific short sequence of amino acids on a protein that acts like a cellular barcode. It tells the ubiquitin tagging system we talked about earlier, I am ready to be destroyed.
That trash barcode.
Exactly. Researchers are exploring ways to engineer artificial molecules like PROTACs that can physically drag a toxic misfolded protein directly to a ubiquitin ligase.
Wait, so they forcefully tag it?
Yes. Essentially, slapping a counterfeit trash barcode onto the toxic oligomer to force the cell's natural wood chipper to selectively destroy it.
It's molecular bounty hunting.
It is highly targeted clearance.
But the ultimate goal of all these interventions, the pharmacological chaperones, the mitochondria transplants, the degrons, the goal isn't to make us immortal, is it? We are still bound by the disposable soma.
Immortality is not the goal? No. Nor is it likely biologically possible. The real goal is increasing health span.
Health span versus lifespan.
Right. Lifespan is how many years your heart beats. Health span is how many years you live free from debilitating chronic disease. Right now, there's a massive gap for many people between when their cellular proteostasis starts to collapse and when they actually pass away.
And we want to close that gap.
We want to tune the network so that the maintenance crews stay active, efficient, and well-funded well into our old age. We want to compress morbidity. We want the microscopic metropolis to run beautifully, the subways to stay clear, and the power plants to hum safely until the very last day.
What an incredible intellectual journey this paper takes us on. I mean, we started with this simplistic old idea of the cell as just a bag of soup, a homogeneous environment where we thought we just needed a chemical sponge to scrub away the big clumps of amyloid plaque.
We saw how terribly that failed.
We did. And through the work of researchers like Nabawi, Dozan, and Vilches, we've traveled down into the microscopic reality. We see the cell now as a highly compartmentalized, dynamically communicating metropolis.
It's beautiful, really.
It is. We've seen walled factories desperately shifting their operations to survive, liquid life rafts turning to solid concrete in the storm, and delicate organelles physically holding hands to share the burden of oxidative stress.
It is a profound testament to the unimaginable complexity of life, and it highlights just how much we still have to discover about the very mechanics of our own biology.
As we wrap up this deep dive into the source material, I want to leave you, the listener, with one final provocative thought to mull over, and this builds directly on that cell non-autonomous regulation we discussed earlier.
Oh, the signaling network.
Exactly. We established that a stressed failing organelle in one tissue, like your gut, can broadcast a chemical panic signal via neuropeptides across your entire body, triggering a premature shutdown in your brain.
Right, the distress flare.
But if that communication network exists, what if the reverse is also true? Could we one day harness this exact same systemic network to send a positive signal?
That is a fascinating idea.
Could we isolate a specific neuropeptide that acts as a youth or repair signal, broadcast from a healthy, robust tissue to rejuvenate the failing organelles in another?
It is a deeply fascinating hypothesis that completely flips the systemic collapse model on its head.
Imagine if, one day in the future, we don't have to drill into the brain to cure dementia. Imagine if simply administering a targeted therapy to heal the proteostasis network in your gut could send a powerful cascading chemical message up the vagus nerve that cures Alzheimer's in your brain.
That would change medicine forever.
A single, systemic, healthy message that tells the maintenance crew in the failing metropolis to pick up their tools, open the bunkers, and get back to work. Thank you for joining us on this deep dive into the microscopic world of proteostasis. We'll see you next time.
Heliox is produced by Michelle Brucker and Scott Bleakley. It features reviews of emerging research and ideas from leading thinkers, curated under their creative direction with AI assistance for voice, imagery, and composition. Systemic voices and illustrative images of people are representative tools, not depictions of specific individuals. Thanks for listening today. 4 recurring narratives underlie every episode: boundary dissolution, adaptive complexity, embodied knowledge, and quantum-like uncertainty. These aren't just philosophical musings, but frameworks for understanding our modern world. We hope you continue exploring our other episodes, responding to the content, and checking out our related articles at helioxpodcast.substack.com.